Hello friends, in this video I am going to explain you the inbred improvement and its utility. Coming to the definition of inbred, it refers to a homozygous line that is derived from an outcrossing or outbreeding population by repeated inbreeding and selection. Inbreeding refers to mating between clearly or closely related individuals. Plant breeding, self-fertilization and sieve mating are common forms of inbreeding. Difference between selfing and inbreeding. The relevance of selfing is to self-pollinated species, whereas inbreeding is to cross-pollinated species. Selfing means pollination of flower by its own pollen, and inbreeding is from pollen of other plant of a line. Crossing is not involved in selfing, whereas crossing is involved in inbreeding. The outcome of selfing is pure line that are homozygous and homogeneous, and inbreeding outcome is inbred line that are almost homozygous and homogeneous. The features of inbred relevance is usually relevant to cross-pollinated crops. Now this term used in self-pollinated species related to rills or recombinant inbred lines. The genetic constitution of inbreds have exactly same genotype, homozygous and homogeneous. The base population in cross-pollinated species, inbreds are developed from heterozygous source, generally open-pollinated varieties used as base population as source. In self-pollinated crops, recombinant inbred lines are developed from a cross between two pure lines. Variation in inbreds are generally pure, hence no heritable variation, but the variation is entire due to environmental factors. Selection. It is ineffective because there is no heritable variation within an inbred line. Adaptation. Inbreds have narrow adaptation due to lack of genetic diversity. Thus, inbreds have poor buffering capacity to environmental changes. Genetic base of inbreds. Inbreds have narrow genetic base due to lack of genetic diversity. As a result, inbreds are more prone to attack of new races of pathogens. The procedure of development of inbred is the produce of inbred is highly attractive and uniform than that of the open pollinated varieties because all plants of inbred line exactly the same genetic constitution, so inbreds have danger of uniformity. Reconstitution. In cross-pollinated species, the reconstitution of inbred with exactly the same phenotype is not possible when heterozygous source is used as base population. The reconstitution is not possible for the primary constituted inbreds, but the secondary constitution of inbreds can be expected to be reconstituted when they are taken from the homozygous source. Improving the characteristics of inbred lines. The techniques are the productivity of inbred increasing for use in hybrid seed production and developing disease and insect resistance inbred lines. The combining ability of inbreds to increase the yielding ability of their hybrids are improved. Breeding methods are pedigree selection, choosing breeding stock on the basis of ancestral reproductive ability and quality are known as pedigree selection. This was given by Lau in 1927. Here why I am discussing breeding method in the only pedigree method in the initial slides means this is a major method in order to develop the inbred lines. Coming to the effect of inbreeding on genotype and allele frequency. As the selfing increases homozygosity, likewise inbreeding also increases homozygosity. Here in this picture you can see that the heterozygous and homozygous balance will be 50-50%, whereas dominant homozygous is 25% and recessive is 25%. In the next generation, the heterozygosity will decrease by 50% and the homozygosity will increase 50% in both the situations, either it may be in dominant or recessive form. Likewise, over the generations, heterozygosity will decline and homozygosity increases. Fewer heterozygotes and more homozygotes, but no change in the allelic frequency. The systems of matings are inbreeders and outbreeders. Inbreeders means many common ancestors or mating of closely related individuals. Outbreeders means few common ancestors mating between not so closely related. Based on genes passed on to progeny, replicates of same gene in parent or not. In non-random mating, the extreme case of self-fertilization, the formulas are given here in different generations. Inbreeding was the process where it may carry two alleles at a locus that are replicated of one or same gene in previous generation. Faulkner in 1960, using Krauss in 1954 terminology, established two sorts of identity to inbreeding. One is alike in state, that means two genes are identical if they are functionally same, that is independent homozygote. The second point is these are identical by descent or by replication, that means identical homozygote. For study of consequences of inbreeding, these are used. Why I am again discussing about the definition of inbreeding here, because this gives the mathematical definition to inbreeding. Coming to coefficient of inbreeding, this is the degree of relationship between mating individuals represented as F. The definition was given by Wright in 1922, where R between uniting gamete, F, referred as inbreeding coefficient. Mellicott in 1948 and Crow in 1954 gave inbreeding coefficient as probability that the two genes at any one locus in an individual are identical by descent. Inbreeding coefficient, or F, is a measure of proportion by which heterozygosity has been reduced by inbreeding. The base population is considered as 0 here. Inbreeding does not change allele frequency by itself, it just increases homozygosity. The coefficient measures the extent to which population depart from the expectations of 2pq. Here HE is expected heterozygosity in Hardy-Weinberg law and H0 is observed heterozygosity. That gives the inbreeding coefficient formula as expected heterozygosity minus observed heterozygosity divided by expected heterozygosity. How can we quantify the amount of inbreeding in a population? That is, with the help of inbreeding coefficient. The probability that a randomly chosen individual carries two copies of an allele that are identical by descent from a recent ancestor. Probability that an individual is heterozygous. Considering F and 1 minus F, the frequency of three genotypes will be like this. The A1 will be represented as P square bracket 1 minus F plus PF, then A1 A2 as 2PQ 1 minus F and A2 as Q square into 1 minus F plus QF. If F is 0, that means the Hardy-Weinberg equilibrium operates. If F is 1, means by inbreeding 2PQ becomes 2PQ into 1 minus F. Inbreeding is mating of related individuals, often result in change in the mean of a trait. It is intentionally practiced to create genetic uniformity of laboratory stock, to produce stocks for crossing like in animal and plant breeding. It is unintentionally generated by keeping small population like is found in zoos. During selection, Coming to genotypic frequency under inbreeding. The inbreeding coefficient, that is probability that the two alleles within an individual are inbreeding depression minus identical by descent. Hence, with probability f, both alleles in an individual are identical and hence a homozygote. With probability 1 minus f, the alleles are combined at random. This is how the inbreeding coefficient is expressed with the different allelic combination in different generations and with different genotypic frequency. Changes in mean under inbreeding. Like in genotype, if A2 is increasing over generation or heterozygosity is increasing, that means A1 will become 0 and the proportion will add to the recessive genotype. Using the genotypic frequency under inbreeding, the population mean under level of inbreeding is related to the mean under random mating by mu f is equal to mu0 minus 2f pqd. This is the changes under inbreeding is calculated with this formula after few generations of continuous inbreeding. This is a graph showing decline in heterozygosity due to inbreeding if SIB mating is done and if selfing is done. The decline in heterozygosity under selfing is quite high or quite rapid as comparing to full SIB mating or half SIB mating. Coming to evolutionary consequences of inbreeding. In large random mating population, most individuals will not suffer from deleterious effect of recessive deleterious alleles. Example in corn. Under inbreeding, increased homozygosity for these recessive deleterious alleles result in reduced fitness. The self and full SIB generation, after certain generations, the proportion of alleles is shown here. Like under self and SIB generation, SIB condition, the proportion at the first generation is 1/4 and in the subsequent generation it changes. In homozygous dominant form, the selfed one will be 3/8 and SIB one will be 5/16, and in another allele to A2 also it remains same, but in A1 A2 it will be 1/4 and 3/8. Likewise, in every generation it changes with a definite amount. Coming to consequences of inbreeding, that is increase in homozygosity, which is shown with the help of number of individuals or with a certain amount of species. The effect of inbreeding is fixation of genotypes, that is locked variability in heterozygosity or fixed as homozygotes, inbreeding depression, reduced fitness, purification of types, increased environmental variability. The response to inbreeding depression in crops, there are certain responses like the high tolerance is shown in onion, rye and cucurbits, moderate tolerance in maize and sunflower, low tolerance in carrot and no loss in all self-pollinated crops. Castor has greater tolerance to selfing. The types of inbreds are first cycle inbreds and second cycle inbred. Apart from these two, there are special type of inbreds like rills, that is recombinant inbred lines, bills, backcross inbred lines, ales, advanced intermating lines, and backcross nils, that is backcross near isogenic lines. Coming to the methods of developing inbred, the first and foremost which is practiced from a long period is selfing. In this, inbreeding of heterozygous population is a common and widely used method. Populations, open pollinated varieties, synthetics, single cross hybrids or double cross hybrids can be used for inbreeding or selfing. Superior plants on the basis of vigor, disease and insect resistance and yield are selected and selfed. Progeny of selected plants are grown separately from the selfed seeds in the next season. Again, the superior plants are selected in each progeny and selfed, then selfing is continued to 6 to 7 generations to get superior inbreds. The main purpose of selfing is to fix the desirable genes in homozygous condition and eliminate the genotypes with undesirable deleterious genes. Production of inbred line through inbreeding. In the first year, selection of plants based on phenotyping and according to purpose from source population are taken. Then in the second year, plants with space planted for selfing, then selection of superior plants or line according to purpose. Then after 6 to 7 generation, the second year step is repeated. Then the inbred developed through half-sieve mating were not superior to those produced through selfing. Coming to the second approach, that is by haploidy, by doubling the chromosome, that is nothing but double haploid in maize, egg cell without fertilization, interspecific cross in potato, polyembryony in lunum pepper and several horticulture crops, semigamy in cotton, nucleus of breadwheat was placed in cytoplasm of Aegilops chordata, that is cytoplasmic transfer in case of wheat, and anther culture in tobacco, wheat and rice. These are all the examples of different crops to obtain the haploids or double haploids to get the inbred lines. This is the process how the haploid plants are developed through anther culture or microspore culture. In the anther culture, the culture vessels are cleaned and they will be made ready for the culture techniques. Then culture selection and preparation of the nutrient medium, sterilization and storage of nutrient medium, selection and sterilization of explants, transfer of sterilized explant on nutrient medium. In the next step, transfer of cultures to culture room for regeneration, then rooting in regenerated shoots, transferring the plantlets to field and hardening. In the later step, confirmation or identification or identifying the haploid plants is also very important. That is nothing but screening. The production of diploid plants through haploids, that is double haploids. In this, haploid plants are taken. Treating with colchicine, double haploids are produced directly, and these will be highly homozygous inbreds. Uses of inbreds for developing hybrids, developing synthetics, composites, second cycle inbred development, gene mapping and linkage mapping. Limitations of inbreds. Inbreds doesn't have heritable variation. These have narrow genetic variability, poor buffering capacity to environmental changes as compared to multiline varieties. Inbreds are highly uniform, so danger of uniformity. Selection is inefficient due to lack of genetic variability. It requires several years to develop reals, bills, airs, nils for crossing, selfing and evaluation and development. Coming to evaluation of inbreds. First process is phenotypic evaluation. The phenotypic performance of inbred themselves. Performance of inbreds is tested in replicated yield trials in different locations and in different years. Top cross test is the second process. It is generally accepted that the performance of top cross progeny of an inbred is a reliable measure of the average performance of all the single crosses involving inbred, and that top cross performance provides reliable estimate of GCA. Third is single cross evolution. It is believed that an important part of heterosis is due to specific combining ability, that is measured by making the single crosses. Then prediction of double cross performance. This prediction from the performance data of single crosses was developed by Jenkins in 1934 by comparative evaluation of prediction based on performance of inbred lines, single crosses and top crosses. Coming to improvement of developed inbreds. Inbreds can be developed through the techniques like selfing, double haploidy and many other techniques. And now I am going to discuss the improvement of already developed inbreds. This is to improve the productivity, quality and resistance to biotic and abiotic stresses. The breeding procedures are: first one is population improvement. In this, inbred improvement takes pedigree selection, backcrossing, convergent improvement, genetic selection, cultivar improvement through mass selection, heterosis breeding, synthetics, composites and others like tissue culture and induced mutation. In population improvement, there are two things. One is intra-population and another is interpopulation. In intra-population, mass selection, modified mass selection, that is Gardner's mass selection or grid method, then half-sieve selection with progeny test or ear to row selection. In this, no replication, or this is called as Hopkins method. Then modified ear to row selection, that is replicated test with Lundquist method. Then half-sieve selection with S-cross, full-sieve selection, S1 family testing, interpopulation, S1 family testing. Up to there, all the techniques belong to intrapopulation selection. The second one is interpopulation selection. In this, reciprocal recurrent selection developed by Comstock and Robinson and full-sieve reciprocal recurrent selection will come. Some of the techniques are discussed here. First one is backcross method. In this, most breeding method for inbred improvement like disease and insect resistance will be developed through backcrossing. In this, a disease-resistant cultivar is taken as a donor and adapted cultivar is a recipient. The F1 is repeatedly backcrossed to a adapted cultivar or recessive one. Then BC1, BC2, BC3 like that. After BC4, one selfing will lead to obtain plants homozygous for the disease-resistant cultivar. Next is convergent improvement. The concept was given by Ricci in 1927. In this, cross involving more than two parents. It is a special case of backcross procedure where a single cross is backcrossed separately to the two parental inbreds. Like this, here three parents are there, ABC. First, AB are crossed by the F1. By taking the F1, it is crossed to C. Then again ABC is a complex hybrid. Then we can take four parents ABCD. We can cross A into B, then C into D. That F1 will be again back crossed to parents, and that A into B and again C into D can be crossed in order to get the double haploids. Coming to gametic selection. When selection operates on gametes on the haploid phase of life cycle, it is gametic selection. This was proposed by Stadler in 1944. Then somatic hybridization. This is particularly the production of cybrid and parasexual hybrid. The steps involved here are: first, protoplast isolation, then protoplast fusion, selection of hybrid cells, culture of hybrid cells, regeneration of plants from hybrid tissue, confirmation of hybridity or hybridization. Then somaclonal variation is In vitro culture of plant cells generate genetic variation. It is known as somaclonal variation. Through genetic engineering, it offers exciting tools to improve specific traits of inbred improved. Example, suitable versions of crygene from Bt have been transferred into maize inbreds and insect resistance commercial hybrid have been produced using such hybrids. In this technique, through recombinant breeding program, by incorporating a gene of interest or a specific segment of interest, a new transgenic plant with the trait of interest can be obtained that will increase the diversity in developed inbred lines. Thus, in a separate part, I am going to discuss about the hybrid variety, which I didn't discuss in my maize video. So, when F1 generation from a cross between two or more pure lines, inbreds, clones, strains, or other genetically dissimilar populations or lines is used for commercial cultivation, that is called as hybrid variety. The hybrid varieties were first commercially exploited in maize. In 1878, Beale reported that certain varietal crosses showed substantial heterosis, that is up to 52%. In 1909, Shull suggested that inbreds should be developed from open pollinated varieties by continued self-pollination. The double cross scheme was proposed by Jones in 1918 and first commercial double cross hybrid variety was proposed by Jones, that is Burlington, in 1922. The concept of top cross hybrid for preliminary screening of inbreds was proposed by Davis in 1927. In 1934, Jenkins provided a reliable method for the prediction of double cross performance. In 1944, about 80% of area was covered by maize under hybrid variety. In 1960, single crosses had become widely accepted in USA. Today, only single cross hybrids are grown across worldwide, along with some composite varieties. In India, intensive research for the development of hybrid maize began in 1952 when Coordinated Maize Improvement Project was initiated in collaboration with Rockefeller Foundation. A large number of inbreds and other germplasm were introduced from USA to India. In 1961, four in maize hybrids, Ganga 1, Ganga 101, Ranjit, Deccan, were released, which were double cross hybrids. In the same year, CGMS based combined Kafir 60 in sorghum and Tift 23A pearl millet were identified. Coming to single cross hybrid, the ratio of male into female planting is 1:1 or 3:1 in single cross hybrids. In double cross hybrids, the female into male ratio ranges from 8:2 or 10:2. In three-way cross, the female to male ratio 3:1 or 4:1. Coming to merits of hybrid variety, they exploit both GCA and SCA components of heterosis. The produce from particularly single cross variety is more uniform as compared to from open pollinated variety. They can be produced both in cross and self-pollinated species. Hybrids are maintained in the form of their parental inbreds, which are grown in isolation and subjected to selection or sibmating. In many self-pollinated crops, hybrid varieties yield 25 to 30% more than the pure line varieties. Coming to demerits of hybrids, farmers have to purchase new hybrid seed every year since they cannot use the next generation of a hybrid crop as a seed. Hybrid seed production requires considerable technical skills. The exploitation of full potential of hybrid variety requires adequate supply of irrigation water and fertilizer and control of weed, disease and insects. Large-scale production of hybrid seed depend on easy emasculation of female parent and on an adequate pollen dispersal from the male parent. In most cross-pollinated species, the requirements of isolation are rigid and ordinarily difficult to fulfill except on large farms. The amount of hybrid seed required to cover the entire area under any crop appears to be impossible to produce with our present seed production setup. If the parents of a hybrid variety differs appreciably for grain quality, the produce of the variety will not be of uniform quality. Coming to important milestones in hybrid breeding, the hybrid varieties have been commercially exploited in most of the cross-pollinated crops, especially in maize. Hybrid varieties successfully in some self-pollinated crops like cotton in India, even tomato, chili, rice in India, China, wheat in USA. In India, hybrid varieties have been released for commercial cultivation in more than 12 field crops. The first hybrid variety in maize were released in 1961. In pearl millet, HB1 hybrid variety released in 1969 by PAU Ludhiana and PHB 1011, BJ104, BK506. These are downy mildew resistant pearl millet hybrids. The first hybrid variety in cotton was H4 developed by C.T. Patel of Gujarat Agriculture University in 1970. Thank you. Hope you liked this video and kindly subscribe my video.